Hello, everyone. My name is Jeffery Amankwah, and the purpose of this research was to discover serum biomarkers that not only detect the presence of CAD in NAFLD patients, but also monitor its progression, offering a better understanding of how these diseases are connected. Traditional biomarkers and imaging techniques for CAD diagnosis lack specificity or are impractical for early pre-symptomatic detection in NAFLD patients. So we hope that this research can be used to create blood tests that have higher specificity and cheaper than the current diagnostic techniques. And so for our materials and methods, the samples were analyzed using LC-MS analysis, and raw data was processed using MS-DIAL and MS-FLOW. Our Excel pre-processing pipeline and statistical analysis were streamlined and run on a custom R-Shiny app that I created using my background in bioinformatics, and visualization was achieved using a separate R program. Metabolite identification was conducted using MS Finder and corroborated using CFMID, which was run on George Mason's high-performance computer, Hopper. And so in our analysis, we saw clear separation between NAFLD and CAD in the positive ionization mode. This visualization has distinct metabolic profiles for each disease. Our analysis identified key metabolites that were consistently upregulated in CAD across all stages when compared to NAFLD. These include metabolites such as sphingolipids, triacylglycerols, and phosphatidylcholines. The upregulation of these metabolites hides their potential as biomarkers for CAD in NAFLD patients. In our analysis of the CAD cohorts, we noticed that the individual stages of CAD, specifically when comparing CAD severity 0 to 1 and comparing 2 to 3, did not show clear metabolic separation. However, when we grouped those two cohorts, the distinction became quite apparent, indicating distinct metabolic stages correspond to the severity of CAD. Within the comparison between those two stages, we observed a significant upregulation of triacylglycerols and a downregulation of prostaglandins in advanced stages of CAD. All metabolites that we found in our progression analysis that met our ASC chromatogram criteria exhibited an upward trend across increasing severity of CAD. This trend supports the notion that as NAFLD progresses to more severe stages of CAD, there are consistent increases in specific metabolites reflecting a progressive alteration in metabolic pathways. In conclusion, our research presents a clear picture of metabolic alterations that differentiate NAFLD from CAD. We've introduced potential serum biomarkers such as sphingolipids, which with their increasing trend may signify advancing CAD pathology. Although there are some limitations to this research, we can validate these biomarkers, which can lead to innovative blood tests for managing CAD and NAFLD patients effectively. I also want to say thank you to Fang, Chiu, Dr. Allyson, Dailey, and my advisors, Dr. Couch and Dr. Chandhoke, for their help with this research.